Yeah, thank you again to our speakers for your very impressive presentation. I guess we have a number of questions in the Q&A, and some of them have already been answered. But anyway, I want to take advantage of my role as a moderator to ask a general question first to our two speakers. So, with the increasing complexity of those spatial multi-omics data, especially like when integrating like diverse data set from various experiments or modalities, although you two talk about primarily the protein like either the cyclic protein type immunostaining, but there are also like spatial transcriptomes and even epigenome from our previous speakers. So when we integrate those type of data, so what are the primary challenges you encountered in your research as a cancer biologist or oncologist? Could you share some practical strategy that you employed for effective data interpretation and hypothesis generation in this context?
You want to go first?
If I'm happy to, but also Katie, you first.
Sure. Oh gosh. So really thinking about, how can the way that I interpret the question is how can these biomarkers be clinically implemented? Because clinicians don't want 20,000 things and so really coming into these the appropriate dimension reduction challenges or the single gene or protein measurements. And I think what it's going to come down to, one is dimension reduction and figuring that out. And I think that there's a lot of basis for that in terms of the gene expression profile scores or the gene scores that have been established thus far that have done a pretty good job at separating kind of the extremes of responses. And I think but the validation of those things across modalities and making sure that it's a clinically relevant assay, I think is one of the greater challenges. And so as we're doing all of this complicated spatial profiling, the fact that we have sequential cuts from those H&E images and the CD8 and the S100 IHC, what quantitation can be done using digital pathology that could be more easily implemented into a clinical setting are some of the questions we're trying to keep in the back of our mind to condense some of these more complicated biologies that are being captured. But I think most importantly, the clinical trials have to bridge with these people driving these technologies and these methods because they need to be assessed in the clinically relevant contexts if you want to actually utilize them. And this is why I very subtly but also importantly highlight that this clinical trial was done in the PD-1 refractory setting. So all of the biomarkers that have been discovered and attempted to be validated, and most of them not necessarily implemented prospectively into clinical trials, aren't quite as relevant anymore for querying biopsies from patients after they've been treated with anti-PD-1 therapies. And so I think context, orthogonal validation to assess scalability and accessibility across institutes and sites is one of are some of the more important things to consider as these things are developed.
Yeah, thank you, Katie. It's a great summary about like the cycle from discovery to clinical trial, clinical implementation, and back to discovery. And Sandra, do you have anything to add?
Well, no, that's a great view on it. I think, from our perspective, we're trying to integrate methods like H&E and IF sometimes on the same slide as we showed. And so being able to do that and having a ground truth to go back to, I think I agree with Katie, having those annotations is really an important part of, I think, of building and discovering new information that builds upon the past. And then in terms of like adding additional methods, so for instance, I think it's important to use excellent methods like Joe was talking about with the products from NanoString. We use GeoMx now regularly, where we do one section of our imaging, then GeoMx on the next section. So commercial methods that are well established are really important to integrate into these kind of more customized, bespoke methods. And then, but like the really kind of out there research where you're trying to do like mass spectrometry imaging on images where you have multiplex antibody imaging, then I think it's really important to work closely with true experts in the field, because everyone knows their own limitations of their platforms and issues about registration, how to get different scales of data together, how to visualize it properly for proper review. So I think all these are really critical points. And I think one of the major challenges of multi-omics is that these methods are so complicated. To get reliable information is not straightforward. So team effort is key.
Thank you. Yeah, I have another question for Sandro. So considering the rapid advancement in those AI-based image annotation analysis, I guess you guys also use a lot of like those type of AI-based tools for like imaging analysis. So I'm curious about your perspective as a pathologist. How do you view the integration of those AIs in pathological image annotation and analysis? Do you believe it's still a distant prospect, or is it on the cusp of revolutionizing clinical pathology, particularly in cancer diagnosis?
Yeah, I think I'm on the side that it's going to be revolutionary, and there'll be a lot of amazing new tools available to pathologists to apply to specimens. I think what I'm very excited about is the ability to integrate both that histologic information, the AI algorithms, with algorithms that take advantage of molecular information, the kind that we're also doing with antibodies. So I think that bridging those two worlds will be very helpful. And I think those two forms of information will talk very, very well to one another if we design the tools properly to do so. Because, for instance, you can, in the H&E world, you can actually pretty reliably identify what tumor cells are. In the IF world, the markers kind of fluctuate. Sometimes, Katie, I don't know if you're like some of your tumors might lose SOX10, right? Oh my, where are my tumor cells, right? So if you have yeah, exactly. So if you want,
we
if
had
you
a
take information back,
CD56 positive, we had a CD56 positive melanoma, which happens, but it's challenging when you're trying to type NK cells.
Right, or half your cells might be SOX10 positive, the other half are SOX10 negative. So if you have an H&E algorithm that supports your tumor calling, but now you can bring in molecular information as well. That's just like one example, I think, of how the two worlds can speak to one another.
Yeah, yeah. And I think I'm really impressed by your like 3D reconstruction, and you showed it's pretty revealing. So do you think like the 3D reconstruction, I guess this is also related to one question in our Q&A. So do you think that the 3D reconstruction can somehow like, reveal some temporal information? For example, after reconstruction, whether it's we can use some like temporal inference or other type of computational approach to, for example, to reconstruct how the tumor invade surrounding tissues, or where's the invasive front involved over time?
Yeah, so like for instance, when Katie described the pseudo-time analysis, we can start to do some of that now with marker data. For instance, that the cell cycle work I was talking about is in essence a pseudo-time analysis where you start at the G1 start and you can like map cells along the cell cycle. What a lot of our analyses miss though is that morphological information. Again, Katie showed that this polarization of molecules. That there's a tremendous amount of morphological information that we leave untapped in the specimens. So hopefully what the 3D will do is allow us to really capture both the polarization of molecular information to a greater extent, but also the sizes of cells. Smaller cells might be earlier in the cell cycle, larger cells will be later. The shapes of cells, the pressure. So there's a lot of, I think, very important molecular morphological information that will be revealed by these 3D analyses. They're still experimental, it's still early. But I'm pretty hopeful that we'll be able to start to like make the case that morphology is actually something that's very important, and that would also help, I think, with some of these pseudo-time analyses as well.
Yeah, sure. Yeah, and I think we try to wrap up this session pretty soon. So my last question to Katie is that, and as we look forward to the future, what specific features or type of information would you consider most crucial for spatial omics to provide for cancer immunology research?
Oh gosh. So from the technical end, I think a lot of the earlier talks did a really good job emphasizing the different perspectives, the proteomics, the transcriptomics, the metabolomics. I would probably take the hot take that we don't have to do it all on one tissue. I think orthogonal validation across multiple sections is good too, especially when you have clinical biopsies. Trying to do too much with one piece of tissue can end up wasting it. So yeah, I think these analysis methods, and I think the importance of the knowledge transfer in order to really push the field forward to make sure that this information is being biologically interpreted and then contextually interpreted for clinical impact. I think that that's probably the my answer there.
Yeah, I guess we have a lot of like molecular level analysis, but it seems that we kind of lag behind from like functional part. For example, for immunology research, if I think maybe the measurement of like cytokines, or if there's a way to spatially mapping those cytokines for phosphoproteins, or even some critical metabolite or metabolic functions, that would be extremely helpful in terms of mapping out the like, for example, T cell activation state or some other type of T cell functions that really matters for the response to immunotherapies.